Good afternoon, if you're on the East Coast. Hi, everybody. We already have over 70 participants, so I'll start with some general comments, and then we can get started pretty soon thereafter. If you have questions to the speakers, you should put them in the Q&A box at the bottom of your screen, and I will moderate the question and answers after each speaker. And beyond that, we can get started. This is being recorded, and we will post the webinar on the Mount Sinai website, the Seaver Center website, by the end of the month. So I want to start by thank you all for joining. It is really a great pleasure to be able to kind of talk about ADNP syndrome and to present some of the things we're doing and to get feedback from the broader community. So I want to thank you all for joining from the Seaver Autism Center. We have quite a few people working on various aspects of developmental disorders, and we can take a look at these disorders from many facets, and particularly within ADNP. We've got quite a few people who are making this a big part of their life work, including Alex Kalevzon, Anna Kostic, Nan Yang, and Paige Seiper, who are all on the panel right now, as well as many other people who are involved in other aspects of ADNP syndrome. And in fact, the list is longer than what you see on the right there. And so we've really decided to focus on ADNP syndrome as trying to advance treatment in the disorder. And I'll talk more about this slide in a few minutes, but basically we take this position, which and many groups do, that there is a way to go from a molecular target, in this case ADNP, or things that ADNP impacts, and use some of the tools, the preclinical tools like human neurons or rodent models, to begin to understand what ADNP in this case does, what a mutation does, and then use that information to think about how to develop new drugs for the syndrome, and ultimately bring the preclinical work into the families, into the subjects, and do clinical trials. And so we have experts working on all of these domains within the Seaver Center on ADNP, and you'll actually hear some of the talks will be, we will talk about both some of the phenotypes of the patients, clinical trials in patients. The rodent model is just now being characterized. Anna's taking the lead on that. Human neurons, which Nan Yang will talk about, and I'll talk a little bit about the molecular aspects of ADNP just to kind of give it some context. So we're way ahead of schedule, but this is the agenda. I'll be speaking first, and as I said, I'll talk a little bit about what we know about ADNP, kind of maybe from for lack of a better word, genetic and biochemical perspective, kind of a high-level view. And then Paige will talk about some of the clinical findings, and particularly sensory reactivity and ADNP syndrome. Alex will talk about clinical trials in ADNP syndrome. Nan will introduce you to stem cell models and how they can be useful in understanding the syndrome, but also potentially in drug discovery and development. Sandra, who leads the ADNP Kids Research Foundation, will speak briefly as well. And I will add a slide before I introduce Sandra about some of the partnerships with other institutions and industry that Anna has put together to get just to show you some of the things we're doing that are kind of outside the Seaver Center in collaboration with others. There will be about 10 minutes of Q&A after each talk. Again, use the Q&A box to ask your questions, and I'll moderate. I will try and keep the speakers on schedule, so I'll let them know if they're coming to the end of their 20 minutes to leave time for questions. And then at the end, there'll be an open panel where we all can kind of discuss as a group questions that still arise. So use the Q&A for questions. Everything will be posted online at the Seaver Center website by the end of the month, all the video. And we have now about 100 participants, and I think the number of participants that are joining us is slowly plateauing. So I think we could get started if that's okay. Okay, so I'm going to bring up some other slides. That didn't work. Luckily, COVID is not over, so we still get to practice our Zoom skills. Just kidding.
Looks like somebody needs more practice, Joe.
Okay. All right. So you should see now a different slide. So I'm going to, this is going to be my talk. Okay, does that look good? Do you see the slide and it's in proper format? Yes.
Yes.
Okay, so as I said, I'll talk about the ADNP and genetics and biochemistry is probably a better way to summarize what I'm going to talk about. So, and I'm going to kind of take a big step back to talk about autism and developmental delay more broadly, at least briefly. So people can understand how the perception of these disorders has changed dramatically in the past 10 years. And as a result, there is a whole another way of conceiving of treatment that is slowly, or even not so slowly, being endorsed by industry. And I think that's going to be very important for the ADNP community. So the Autism Sequencing Consortium is something that we put together over a decade ago. It was started by these two gentlemen, one who's since retired, one who came out of retirement. They signed, as you may remember, the America Recovery and Reinvestment Act. And a small amount of all of that money, really quite a small amount, a few percent, went to science. And it went to new projects that had never really been supported before. And one of the things that had just emerged after the full sequence of the human genome were methodologies where you could look at everybody's DNA, sequence every base, and find rare mutations in a fairly cost-effective way. And so this was our first meeting, second one, actually. The first one got snowed out in 2010 here in New York Academy of Medicine. And we put together the idea that if we want to find causes of autism using this new technology, then we should all work together as an international consortium, because historically each group worked on a few samples that they had, worked with a few families, and it was never enough to find anything. So we said, can we kind of proactively decide that we're going to all work together and try and solve the problem of autism and intellectual disability from the genetic side as a group. And in 2012, some of those lead groups put out 4 papers using this new technology. And as I said, this one over here was supported by the ARA Act. These 3 were supported by the Simons Foundation. But collectively, we sequenced 1,000 individual families with autism or intellectual disability, and we found 9 genes. So suddenly, autism was considered this very, very heterogeneous and complicated condition. The causes were not known. And suddenly we were able to say that these 9 genes accounted for some parts of autism. So more to the point, if somebody was carrying a mutation in one of these 9 genes, that probably was the major cause of their autism. So suddenly, for a small number of individuals, we could say this is the cause of the autism and intellectual disability in those individuals. The ASC continued to put together more samples. This was the first kind of ASC-wide paper where we had over 12,000 samples sequenced, 3,800 individuals with autism and developmental delay. Sylvie Dubé of the Seaver Center was the first author on this paper. And suddenly we found now not only 22 genes that were contributing to autism, but those genes appear to be found in about 5% of individuals with autism, which doesn't sound like much, but we went from not knowing anything about a specific cause of autism to now being able to say in 5% of individuals with autism and developmental delay, these genes are mutated. Okay? And these genes, this is the genes with the strongest evidence. This is somewhat less strong. This is somewhat less strong. These are alphabetical, but you can see that ADNP was discovered as a highly significant autism intellectual disability gene in this large study. Yeah, my next slide actually, it's a little blue circle around the word, around ADNP. We continue to get larger and larger samples. This came out actually now 2 years ago in 2020. And now we have a much larger sample, 30,000 individuals sequenced with this technology. And now we have 102 genes that are contributing to autism, intellectual disability, and developmental delay. And this is one way we plot those genes. This is where they are on the human chromosome. This is the evidence that this gene is an important gene. And the higher up it is, the stronger the evidence, right? And so what you can see again, now we have 102 genes that are contributing to autism and intellectual disability when mutated or developmental delay. And again, ADNP, there's my little blue circle, ADNP is at the very, very top of this list, right? And that means both that it's a highly penetrant gene, in other words, that it's a gene that accounts for most of the risk in the individuals with mutations, if not all of it, and it's a somewhat more common cause of developmental delay than some of the other genes. So a gene down here is a real gene, but it's probably much rarer than ADNP in terms of how often you'll see it in the developmental delay population. Just about a month ago, we put online on an online preprint server our newest study, now with 63,000 individuals. So it just gets bigger and bigger. And again, an international consortium, people are contributing samples from all over the world. And we now have about 185 genes by the same criteria I showed you earlier. These are the top 71. And what you can see here is ADNP is right up there, right? So what does that mean practically? It means that ADNP is one of the more significant causes of developmental delay, of autism, of intellectual disability when mutated. We have a good sense, and I'll show you in a few minutes, what kinds of mutations lead to the syndrome. And we can say, with some, I think, and it's important to be able to say this, with real data, that it's not an ultra rare syndrome. And so we think that brings in more people in industry that are trying to look at, they want to make sure that if they develop something, there's a patient population or a family population. And just to kind of talk a little bit more about this new perspective on autism, we took Brian Majani, who is in my group and is about to become an independent faculty member. He took data from a national cohort, which has the advantage of that it's unbiased in terms of who's being sequenced, and was able to show that with current knowledge, we can already provide a genetic diagnosis in nearly 30% of individuals with autism and developmental delay and intellectual disability. And that number will go up as we find more genes. So again, in 2010, we didn't know the cause of any autism. In 2012, we found 9 genes. By 2014, we could explain 5% of autism and intellectual disability developmental delay, and now we're up to 30%. And that has really changed the field, both in the academia and in industry, because now people are thinking about these conditions as rare genetic diseases, right? So it's no longer how do you treat intellectual disability, or how do you treat language delay, or how do you treat autism. It's that as well, but it's also how do you treat the specific rare genetic disorder where there is a mutation in a specific gene. And I'm sure you heard this, that there are many estimated 7,000 rare genetic disorders known so far. Most rare diseases are of genetic origin. When you put all of the rare disorders together, it has a major societal impact, primarily affects kids. And there are special pathways, incentives within both commercial and research to make efforts and regulatory as well, very importantly, to make inroads into rare disorders. And there are quite a few, as I'm sure you've heard, new therapeutics that are actually in the rare disease space, and they've proven to be not only very effective, but also provided a good return on investment, which is what's important for engaging industry. So, there is a shift in developmental delay syndromes to go from treating them as a series of behaviors that we certainly want to treat and will continue to treat, but also to treating them as rare genetic disorders, where you can maybe come up with a treatment that's specific for the rare genetic disorder. And so what we do at the Seaver Center, and this is the slide I showed you a few minutes ago, I'll just kind of go through it in more detail now, is we have kind of 4, if you will, levels of internal expertise that can help drive things forward. First of all, through the ASC, we can identify the top targets for intervention, the genes, and then we could do work on molecular biology or biochemistry to ask questions. In the case of ADNP, what's the function of ADNP? What does it do in a cell? And how do the mutations change that function of ADNP? And hopefully those kinds of studies could suggest a treatment direction. If you see something that's going wrong in the cell, you might say, okay, that's going wrong. Are there ways I can correct that? The second level is on human neurons. So as Nan will talk about, there are methodologies now to take blood samples and turn them into nerve cells and basically study nerve cells in a Petri dish from an individual with ADNP syndrome. And so now you can ask, what does ADNP do in neurons or nerve cells? What's its role in nerve cell development or function? How do the mutations affect this? Is there a change in the nerve cell function or development? And are there ways to reverse this, right? So you can work in a Petri dish and begin to see whether you can turn things around to, if you see a change, can you put it back to where it should be? And then, if you see these kinds of changes caused by the mutation, then you can use that information to test a treatment. So you can say, okay, the nerve cell looks different than it should. I put on a treatment and it went back to what it looks typical now. So that's a test. But you can actually also use these nerve cells to identify treatment. So you can actually screen compounds, and I'll mention that a little bit at the very end, and Nan will mention it too, on a large scale to find compounds that are effective in reversing a change in nerve cells that's caused by the ADNP mutation. The other tier we work on quite extensively is genetically modified rodent models. So these are, for example, mice and rats. In this case, it's mice that have a mutation in ADNP that mimics a clinical mutation. And now you can ask, what does ADNP do in animals? What's its normal function? How do mutations affect that normal function? And again, if you see some change caused by the mutation, are there ways to reverse this, right? And certainly, if you found something that was positive here or here, you could test it in your genetically modified mouse model and say, oh, look, I see a change, and this compound, this drug, this gene therapy reverses that change, right? So it can be a very useful preclinical test for a therapeutic approach. And then ultimately, of course, you want to understand what ADNP does clinically. And again, what can be done to treat that with existing knowledge. But for the future, how can we take some of these things that came out of the preclinical studies, which are all of these, and use them to treat ADNP syndrome? And so, and some of these things, like particularly if there's a compound here or a treatment here, bringing it to patients is something that actually pharma and biotech do very well. And so there's also a very important place for industry in this pipeline. But what we can do in academia is quite a bit of this, and even some of the transition from preclinical to clinical. We have experts on that at Mount Sinai and elsewhere. But this is how we see this drug discovery and development pipeline. And Anna Kostic, who's won't be presenting today, she's been leading this effort for ADNP syndrome, putting together the work of the molecular biologists, biochemists, the work that's going on in the stem cells. She's going to lead the characterization of the new mouse model that the ADNP Family Foundation supported. And you'll hear both about phenotyping from Paige, but also about clinical trials from Alex. Even just seeing individuals with ADNP syndrome and asking about the clinical manifestations is a critical part of a drug treatment pipeline. If you don't have your clinical endpoint, you won't be able to do a clinical trial. So just seeing families and bringing them in and understanding the phenotypes tells us what are some of the things that we could use to target as an endpoint for a clinical trial. So, like I said, I will talk more about what I'll loosely call biochemistry. Genetics I've talked about. I'll talk a little bit about chemistry, but just at a very high level. So one of the things that genetics tells us from the same paper that's now online is that ADNP, the mutations in ADNP work by a single mechanism. And that's important because there are different kinds of genetic disorders where every mutation is different, right? But we know with ADNP that virtually every example we know about is a protein truncating mutation or a deletion. And almost none of them are what we call missense mutations. But you can see that some genes, almost everything is a missense. So missense mutation is a certain kind of mechanism if it's only missense. But genes that are only or almost exclusively protein truncating, where the gene actually cuts the protein in half, or tells you something about how the disorder is best modeled. So it is a loss of function or a haploinsufficiency syndrome. And this graph tells you that almost everybody with the syndrome, if not everybody that we looked at, has a de novo mutation. In other words, they didn't inherit it from a parent. And that's important because that tells you that these mutations are causing virtually all of the disorder. Right? If there's a mutation that can be inherited from a relatively healthy individual, then you know that there's other things going on that produce the final manifestation of the disorder in the child. When you have no inherited mutations, in other words, it's all de novo, that tells you that it is the appearance of that de novo mutation that accounts for virtually everything you're seeing in the child with ADNP syndrome. So we learned two things from genetics. One is that it's a haploinsufficiency, the loss of one functioning copy, and it's being driven, and those mutations are the primary cause of the disorder. And again, pharma wants to hear that. They don't want to hear that, we think, we're not sure. They want definitive evidence like this that says it's a haploinsufficient syndrome and it's highly penetrant. In other words, most or all of the risk comes from the mutation. The other thing we know about ADNP syndrome on a somewhat broader level, and Nan, her work follows up on this, is that ADNP is involved in neuronal maturation, nerve cell maturation, and nerve cell differentiation. So nerve cells, like all cells, start as a single ball, and then they have to spread out and they make connections. So they mature and then they differentiate. They become very, very elongated and they connect with the nerve cells. And this is work that Matt Lally at the Seaver Center had done before he came to the Seaver Center. He's now continuing this work. And he looked at several genes that are implicated in autism and developmental delay, including ADNP. And he showed that a subset of them are directly involved in nerve cell maturation and differentiation. And furthermore, that when he reduces the levels of ADNP, which is if something is a haploinsufficiency syndrome, then to model it or to mimic it, you reduce the expression. So he showed that when he reduces ADNP in these nerve cells, it delays the maturation and differentiation of the nerve cells. Okay, so that it's in a petri dish, but it's already telling you that something about the development of nerve cells and probably also the brain will be impacted by ADNP mutation. On a very broad level, it turns out that ADNP also regulates the expression of many other genes, and that's that's a complication, right? In other words, it's not just ADNP that's changing; it's all the genes that ADNP, or many of the genes that ADNP regulates, may also change. And it's been shown, again, that if you reduce the levels of ADNP, as you would see in the syndrome, you affect the changes in these downstream genes. These are 3 papers. There are many more. Not many more. There are several more. And I'm going to talk a little bit about 1 or 2 of these on a very high level. But the important thing is that Poxy, together with ADNP, I don't know if you can see it, but it's right there. It affects how DNA is packaged. It affects how genes are expressed. ADNP affects how, again, DNA is packaged, how DNA and RNA interact with each other, how a protein called CTCF, which I'll talk about very briefly later on, interacts with your genomic DNA. And ADNP is a part of modulating that whole process. And here's yet another example of a complex called CHAP, which I'll talk briefly about. CHAP stands for CHD8, ADNP, and HP1, and showing again that it's involved in how the DNA is structured, which in turn regulates the expression of genes. So we have 20,000 genes in our bodies, in our DNA, and they all have to be turned on and off in very specific ways. Your eye needs to have crystalline in it to be able to be transparent. If crystalline didn't turn on or something else turned on in your eye, it would not be transparent, right? So all those 20,000 genes have to be turned on and off in very, very carefully orchestrated ways. And ADNP turns out to be one of these regulators of all these other genes. So this is a recent paper, came out at the end of last year from some colleagues and friends. And actually they're working on POGZ, which I'm going to come back to in a moment. This is this CHAP complex, CHD4, ADNP, HP1, and POGZ turns out to be part of it. And they show that there is a balance between POGZ and ADNP, which determines whether this particular gene here, for example, of those 20,000 genes in this cartoon, whether this particular gene is expressed or not, whether there's activation or repression of expression. And what they show very elegantly is that Poxy and ADNP can compete for the same site, or they can co-localize, for the same site. And depending on the levels, that's what's going to turn the gene on or off. And if you add ADNP, you can inhibit the Poxy function, and that you really need a balance of the two of them. And so again, if you're missing some, that balance is no longer maintained, right? So, and I'll come back to this in a moment. This is again about the CHAP complex, and again talking now about differentiation of cells. And so if the CHAP complex is functioning as it should, then cells, very early in development, cells retain what they're supposed to retain. It basically keeps them shut down until they're ready to be activated. But if you disrupt this complex, either by disrupting CHD8, HP1, or ADNP, then you get abnormal gene expression. So the cells that are supposed to be sitting there quietly in development suddenly become activated and differentiate rather rapidly. And when they do that, it interferes. If they were going to become nerve cells, it interferes with them becoming nerve cells. So this complex, which includes ADNP, is beginning to tell us a lot about the function of ADNP, but we also have to look at the other members of the complex to understand the whole thing, right? So we have to look at CHD4, Poxy, HP1. So to kind of put that into a bigger context, ADNP is part of this CHAP complex, CHD4, ADNP, HP1, but the HP1 itself forms multiple complexes, and they are all critical in brain development. So these are proteins or genes that bind to HP1, ADNP, CHD4, you've heard about that, there's HPN1 alpha, but there's also EHMT1, EHMT2, Poxy, which I mentioned previously, CHAMP1, which I'll show you in a minute. And it turns out that many of these genes are associated with developmental disorders, neurodevelopmental disorders. In other words, there's a cluster of genes that work very closely together, and mutations in pretty much any one of them, not any one of them, but many of them are already known to be associated with developmental delay syndrome, right? And I'll show you that now. I'm wrapping up in a minute. So this is the slide I just showed you. These things all interact with each other in different ways, right? So many of these gene names, they don't mean anything to. I mean, you all know, you've heard of ADNP, and some of you have heard of CHAMP1. These are all different gene names. So each of the 20,000 genes in our genome has some name like this, and they're hard to keep straight. So many of these are critical in brain development, and then I said many of them are already associated with neurodevelopmental disorders. So this is the slide I showed you before, where I showed you the genes we're finding in the current ASC Autism Sequencing Consortium freeze. ADNP, as I said before, is amongst the top genes. Here's Poxy. So Poxy, as I showed you before, binds to ADNP. Here is CTCF, which is the gene I mentioned before, that is also part of the Poxy, part of this whole complex. EHMT1 and EHMT2, EHMT1, I don't know if you can see it. This is another study by a group in the UK looking specifically at developmental disorders broadly, not just autism developmental disorders. I know there's an arrow pointing to something useful, but I can't see what it's pointing to. But there's CHAMP1, and I know that EHMT1 is there too, right? And Poxy. So basically, these are genes that are being discovered as drivers, causes of autism, intellectual disability, and developmental delay in our study. And these are genes that are being discovered as, when mutated, as being causal for developmental delay syndromes in the UK. And obviously, there's a huge amount of overlap. There's ADNP, top gene. Some of the other ones are, but you can see that in this really quite small network of proteins, right? We have 20,000 genes and even more proteins. A very high fraction of them are already known to, when mutated, cause developmental delay syndromes. And so studying not just ADNP, but studying the complex is going to be a great way, using kind of biochemistry and preclinical studies, to shed more and more light on the function of ADNP and understand, what it's supposed to do and what might be going wrong. And so we have, we have collaborations with many groups working with many of these genes, including Poxy, we also think that the family foundations, and there are efforts in this regard, could get together and kind of work collectively around these genes that actually function together, because a win for one could be a win for all. Right? And we did invite the CHAMP1 community to these talks because it's one of the, so CHAMP1 is this gene over here. As I said, it is a gene that's been discovered as a strong cause of developmental disorders. And we were approached by the CHAMP1 Family Foundation, and we decided it would be worth also studying CHAMP1. Here, this is a clinical study that's about to go live, hopefully very soon, because if we learn about CHAMP1 or POGZ, we will learn about ADNP. And if the CHAMP1 Family Foundation and the ADNP Family Foundation, the POGZ group, would coordinate, then I think it's to everybody's benefit, right? So we do think that, not looking at every gene, but looking at genes that function together gives us huge insight into the function of the genes, and is a way to leverage all the studies to make clinical advances in each of those disorders. So I'm going to stop there. And thank you again from the Seaver Center. I'm going to end the show. I'm going to try not to lose my voice. I'm going to stop sharing. And I'm now going to open the Q&A. Just look at the time. Yeah, good. So I'm going to, all right. So unfortunately, I didn't look at the chat while I was presenting. It sounds like my pointer was not visible. We'll try and make sure that's not a problem again. There's a question about recordings, and the answer to that is yes, we are recording this, and it will be posted on the Seaver Center website by the end of the month. Yes, so that answers two questions. And then there's a question from Tamara about systematically testing every child with autism and developmental delay using the methodologies that I showed you. And that is one of our goals. So we are working with a larger group of people, including with genetic testing laboratories, to try and change the standard of care, right? Because many times when somebody has a child with developmental delay, they're not tested for these genes or for any genes. And the problem with that is then we don't know how many of these kids have these mutations. When I go to, the ADNP Foundation can tell you this too, when they go to a pharma, a biotech, the pharma says, well, how many cases are there? And if you show them your wonderful registry and there's 300 or 500, they say, well, that's nothing. We know there's many, 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 many more, because I can show you that about 1% of the people we sequence has an ADNP mutation. So it's critically important for the community, for the medical community, and for the research community, and for the family foundations, to move us forward with genetic testing, so that everybody can know, we can know how frequent these mutations are. It will be an impetus towards developing novel treatments. And of course, it's an opportunity for families to learn from each other, but also to participate in clinical trials, like what Alex told you about. Right? If you don't know if your child has ADNP syndrome, you won't be participating in the family Facebook group, and you won't be learning from other families, and you won't be pushing people like me to work harder, and you won't be pushing the NIH to put more resources into it. You won't be able to go to pharma and advocate if you don't know that, and you won't participate in the clinical trials. So that's a long-winded answer to that question. Maybe I'll take just two more. Remember that we're also going to have an open Q&A at the end, so we can come back to some of these things. Sandra is asking the toughest questions. So one about the connection between the three genes. There are multiple connections, and Sandra asked me if I could explain it easily, and the answer is no, I can't. Easy. That's actually an easy thing to answer. No, I can't. But we can take that offline. Let's see. In what phases of the development of the child does the deficiency of ADNP protein affect neurodevelopment? We don't really know that, right? So obviously, the brain is developing. If there's something goes wrong there, maybe it's harder to reverse, right? It happens if ADNP is functioning throughout life, and anytime you put it back, things get better. That would be great. We don't know that yet, but obviously, for most neurodevelopmental disorders, you want to try and intervene as soon as possible. But at the same time, when people do work in mice, they show that when you turn genes back on later in life, things do get better. Maybe not 100%, but maybe 80%. So international collaborations, there are now about 50 groups that participate in the ASC. We're trying to develop we've created an international network for some other disorders. We'd love to push to help contribute to an international consortium for ADNP. Somebody's asking about racial ancestry and racial backgrounds and these mutations. And from everything we've looked at, there's not really much a difference across ancestry. Remember, these are de novo mutations. They're not inherited. They occur spontaneously in genes that we all have that are not tolerant to mutations. And so, every time we've looked, we see these kinds of mutations come up irrespective of somebody's racial or ethnic background. The term I use is ancestry, which is a neutral genetic term. And then, of course, one of the questions that's come up here, and maybe this is for a later conversation, why are there differences, in terms of the phenotypes, right? Why are some individuals more affected and less affected? And that's something we don't know the answer to yet. We don't think, as I said, we don't think it's because there are other major genes contributing, but we can't fully exclude that. But I think that's the next big thing to begin to understand. And Mark asked about dominant negative. Yes, so good question, very, very, very good question. But probably beyond the scope of this talk. But one of the interesting things, complexities about ADNP is that it does not undergo, many times when we have a mutation, the mutant product goes away. ADNP is one of those rare examples where the mutant product does not go away. And so, you can consider models where that is is the cause of the disorder. So it's a haploinsufficiency syndrome, Mark, in the sense that missing a copy, if you will, or having a PTV is the cause of the disorder. But you're right, we cannot formally exclude, and I was trying to nuance that a little bit. But because there's an expressed truncated protein, obviously, from your work and other people's work, there is a possibility of having a dominant negative effect. All right, so I think that's all we can do right now. I'm going to welcome Paige Seiper. So Paige is our chief psychologist here at the Seaver Autism Center. She's an assistant professor in the department, and she has done lots of very, very elegant work on phenotyping and also developing new tools for phenotyping individuals and biomarkers. And she'll talk about sensory reactivity in ADNP syndrome.
Thanks, Joe. So first, thank you for having me. I'm really excited to get to talk today about sensory reactivity in ADNP syndrome, an area I'm passionate about, and as you'll see throughout my talk, why this is so relevant to ADNP syndrome. So I want to first just disclose that one of the measures I'll be talking about today, the Sensory Assessment for Neurodevelopmental Disorders, which we also refer to as the SAND, is a measure that was developed here at the Seaver Center by myself and a colleague, and has been licensed by Mount Sinai to a publishing company. So several key questions that I'm hoping will guide my talk today. I really want you all to have a sense of what sensory reactivity is and why it's important, how we measure it in syndromes such as ADNP syndrome, ranging from behavioral assessments to measures looking at neurophysiology. I also want to talk about and really highlight what we've learned so far about sensory reactivity in ADNP syndrome. I'll include both recently published work as well as several data sets that are not yet published. And then I will talk a little bit just about our future directions, what we're hoping to learn in this area. So to give context to my talk, I want to start by everyone taking a moment to think about the fact that we all have our own sensory preferences that naturally in our everyday lives, we've learned how to regulate. So some of us enjoy or benefit from ambient noise while we're working or engaging with others. So we work well in a coffee shop or with music on. Other people need a quiet space, so they might prefer to work from a library or in a single office. Some people want a room to be bright when they're working, whereas others prefer sunlight or light from a lamp. Some people love the feel of sand and the sound of the ocean and enjoy being at the beach, while others would prefer to be at the pool or would prefer neither. We also make adaptations naturally throughout our lives. So on a sunny day, we might benefit from working with the natural light, but on a dark day, we maybe aren't as comfortable because we have to have our bright lights on. After going for a run, your temperature of your shower that you choose would definitely be different than how you set it in the morning on a cold winter day. So for most of us, we're able to identify our sensory preferences and modify our environments to make ourselves as comfortable as possible. But what we see in many individuals with autism and related conditions are sensory abnormalities. Many individuals aren't able to describe what their sensory preferences are, or are unable to modify their environments independently. And I think one of the really exciting aspects of the work that we've done at the Seaver Center is we've really figured out ways to, as objectively as possible, understand an individual's sensory preferences at many different levels of functioning. And we're starting to think about ways that we can then use that to personalize treatment approaches. So in 2013, when the Diagnostic and Statistical Manual of Mental Disorders, the DSM-5, first came out, this was the first time that sensory symptoms were included in the DSM as a core feature of autism. Now there's two core domains of autism. What we refer to as the A domain is social communication and interaction. An individual has to meet all three diagnostic criterion in this domain. In the repetitive and restricted interest domain, 2 of 4 criteria must be met. And this is where sensory abnormalities fall. So an individual does not have to have sensory reactivity differences to meet criteria for autism, but the prevalence is extremely high. And the way that sensory reactivity is defined in the DSM, they separate it into 3 categories, which I'll show you. And when we get into interpreting the data, it's really important to have an understanding of each of these distinct sensory reactivity phenotypes. So hyperreactivity refers to an adverse response to sensory stimuli. So this might look like individuals who avoid or are bothered by bright lights, not liking the texture of certain clothes or the feel of certain textures on their skin, covering ears in response to everyday sounds. Hyporeactivity is essentially the opposite of that. It's an indifference or an under-responsiveness to stimuli. So an individual who might not notice a car passing by or something moving by them, or individuals with high thresholds for pain or temperature, or those who might not notice or might show a delayed reaction to the sound of an alarm, for example. And then sensory seeking is an excessive interest. So visually inspecting objects near one's eyes, mouthing objects, seeking the feel of objects against their skin, putting objects that make noise close to their ears. Now, I think one of the most interesting things about sensory reactivity is the way that we can translate this to personalized treatment, right? So clinically, the way that I would recommend to treat hyperreactivity, an individual who is bothered by certain environments, would be very different than what we would recommend for an individual where there are safety concerns because they might not notice the sound of that alarm. Sensory symptoms also are really now considered a common feature of genetic syndrome. So there's this growing body of literature describing sensory reactivity phenotypes, including out of our group where we've described differences in Phelan-McDermott syndrome, FOXP1 syndrome, DDX3X syndrome, and I'll of course talk about our ADNP results. It's also really important to think about the fact that sensory reactivity can be measured at different levels. So when we're thinking towards clinical trials, this is really important. We can translate some of the tasks and some of the metrics that we're looking at in patients to animal models. And so this is certainly an area of real interest. We also think that sensory symptoms can change. When we think about higher level social, cognitive, and language skills, these are skills that often are taught, whereas we're not taught our sensory preferences, right? These are things that are innate to us, where environmental modifications can make us more or less comfortable in a given setting. So I'll focus on the tools that we're using here at the Seaver Center to measure sensory reactivity, specifically in ADNP syndrome. In terms of brain-based measures, we're looking at high-density EEG. So you'll see these caps where there are 128 electrodes that we're using to collect data on aspects of visual processing and auditory processing. You'll see the second image is what we call a visual evoked potential. So we're able to look at the earliest stages of visual processing, the first 100 milliseconds of a response. And then the last figure is an image of our eye tracker. So we're able to use eye tracking to look at visual attention, for example. In the behavioral domain, we're looking at the sensory profile. So this is a parent-reported questionnaire. And then we're using the sensory assessment for neurodevelopmental disorders, or the SAND, which gives us two critical pieces of information. It offers us an observational assessment. So you'll see this image of a lot of fun sensory toys where we're actually able to prompt sensory behaviors. And we're also doing a corresponding caregiver interview where we're asking parents the same questions that we're coding as clinicians and breaking it down into the hyperreactivity, hyporeactivity, and seeking domains across visual, tactile, and auditory modalities. And I just want to highlight a few key aspects of the SAND. So when we developed the SAND, we really had the benefit of learning from some of the gaps in the field. So of course, the largest gap was the fact that the DSM-5 came out, but there were no measures to quantify symptoms in an objective way using observational and interview formats. And the second thing was, from a research perspective, we've really struggled with how do we combine data from individuals who are given different types of assessments based on their age or based on their language ability. And so it was really important for us to develop a measure that had continuous variables where we could look across a population and across individuals over time, looking at the exact same numerical values, where higher scores meant the same thing. And the total possible score was also the same. We wanted a measure that clinically could personalize treatment planning, that could measure change in response to treatment and over time, but that also would hold significant value in the area of research and clinical trials. This SAND is appropriate for individuals regardless of level of functioning. Individuals can have few to no words. We also developed the SAND with straightforward administration and scoring. So as best as possible, we wanted to make this the most objective tool that we possibly could. We also benefited from literature around 2 gold standard autism diagnostic assessments, which really showed that the combination of a clinician-administered interview and an observation produced the most robust diagnostic picture. And we wanted to make sure this was low cost and low burden that could be used across disciplines. So here's an image of some of our beautiful ADNP models. And I really want to highlight that the data that we're collecting is not easy, but the fact that we're able to collect such objective data from so many individuals with ADNP syndrome has been remarkable. And it really shows that connection between the families, between your children, and between our group, who's all really been motivated towards that same goal with the excitement when we actually get the data. And because of that collaboration, we've been hugely successful in obtaining really objective data. And I'll show you some of those results. So jumping into the results, the first study I'll share that was published this year about sensory reactivity in ADNP syndrome, we really were asking the question of, one, what does this profile of sensory symptoms look like in ADNP syndrome? And is it related to age? Is it related to sex, to the presence of autism, to IQ? And what we found were high levels of sensory seeking across domains. We saw high levels of hyporeactivity in the tactile domain, driven by a high threshold for pain and temperature. But interestingly, we saw that there was no difference based on these other variables that I mentioned. In particular, half of our sample, and I saw this was a question earlier in the chat, in our sample of 22 individuals, half of the sample, 11 of them met criteria for autism. And I think it's important to highlight that we see autism symptoms in many individuals with ADNP syndrome, but there's certainly a subset whose social responsiveness exceeds what we often see in individuals with autism. So when we think towards clinical trials, we're thinking, how can we figure out what clinical outcomes we want to assess and what our outcome measures should be? And one of the questions is, are the symptoms we're looking at prevalent in the majority of individuals with ADNP syndrome? And in the case of sensory reactivity, the answer is overwhelmingly yes. So when we look at this figure, I'm just going to explain a couple interesting things that came up. So if you look in the middle at the histograms, the top row is comparing individuals with ADNP syndrome to typically developing controls, and these are all age matched. And in the bottom group, it's showing individuals with ADNP syndrome compared to those with what we call idiopathic ASD. So autism, where in the absence of a known genetic cause. And what we see, you'll see each of these lines is a standard deviation. And what you'll see on the top is that on average, almost every individual with ADNP syndrome fell over 3 standard deviations above the TD mean on total score on the SAND. And yet total scores for ADNP syndrome and idiopathic ASD actually looked quite similar. But when we broke it down into these more discrete domains, what we saw was that the profiles were actually quite different. So when you look at the figure to your right, what you'll see is that anything, I guess to explain how the Z-scores work, a value of +1 would be 1 standard deviation above the mean, -1 would be 1 below. So a couple of things I want to highlight. When we look at sensory seeking, we see between 2 to 4 standard deviations above the mean in visual, tactile, and auditory domains compared to controls. And even compared to idiopathic ASD, we see over a standard deviation above the mean in tactile and auditory seeking. Whereas individuals with ADNP syndrome actually showed less auditory hyperreactivity than those with idiopathic ASD, and showed at least 1 standard deviation above the mean on average on that tactile hyporeactivity that I mentioned compared to idiopathic ASD. Another thing just to visually highlight here is that the data is quite evenly distributed within the ADNP group. And the data actually looks really good to think about translating it into developing ADNP specific norms. So not just how does this individual compare to other individuals with autism, but how does this individual compare to other individuals with ADNP syndrome? We then looked at several other measures where we capture sensory reactivity. So we looked at the short sensory profile. Now, the short sensory profile combines under-responsiveness or hyporeactivity and sensation seeking into one domain. And you'll see when you look at the combined column that 90% or a little over 90% of parents or probable differences in this area. We also saw 82% showing overall total score sensory reactivity differences. We also looked at the ADOS, the Autism Diagnostic Observation Schedule, and the Autism Diagnostic Interview. These are two gold standard autism diagnostic assessments. For those of you who have come in, you're familiar with these. The ADOS is about a 45-minute assessment that's administered by a clinician, and there's one question about sensory seeking behaviors. And what we saw was that over 86% of cases showed at least one sensory seeking behavior during this brief assessment, and that 100% of parents reported both current and past sensory seeking behaviors on the ADIR. And that's compared to other aspects of sensory reactivity that were measured, such as sensitivity to noise, where we saw 57 to 66%, and other hyperreactivities where symptoms ranged from 23 to 42%. I'll also note here that when we look at current versus ever scores, this is suggesting that scores are staying stable in the sensory seeking domain, but there does appear to be some improvement in the hyperreactivity domain over time. So what does sensory seeking look like? So I'm going to show some examples of what it looks like. So seeking out sounds or the feel of objects. And in looking through these videos, I think one of the things that was really interesting is seeing across different children some of the exact same behaviors with certain stimuli. I'll show you one more example of some other types of sensory seeking that we see. I think one of the other benefits of the way that we're measuring sensory reactivity, having the benefit of developing a tool after understanding syndromes such as ADNP syndrome, is that we're able to capture aspects of sensory reactivity, such as mouthing of objects, where certainly we've seen many kids where the mouthing of objects actually helps them engage in the tasks at hand. And so this is what we call satisfying sensory cravings. All right, we also looked across different age ranges in this same paper, and we saw no difference in these various age cohorts. And this is something that we're really interested in looking at, in changes in sensory reactivity over time. In this case, this is looking cross-sectionally at the data. All right. So now to jump in about and follow up on some of the work that Dr. Buxbaum shared, and now we're getting into all the unpublished data. One of the things we were interested in looking at was, how did the ADNP phenotype compare to CHAMP1, where we know there's a relationship with ADNP, and Phelan-McDermid syndrome? And very interestingly, now I should mention all the CHAMP1 data was collected during COVID remotely. So this is all comparing just the parent reported data on the SAND. And what we saw was that the ADNP and CHAMP groups showed this exact same profiles on the SAND with somewhat greater seeking reported in ADNP. Whereas in the PMS group, we saw this very clear high level of hyporeactivity. With greater seeking relative to hyperreactivity. On the SAND, when we looked at the modalities, so visual, tactile, and auditory modalities, we also saw the same pattern where the CHAMP1 kids showed the same pattern of symptoms, with the greatest number of symptoms reported in the tactile domain, similar to ADNP syndrome and different from PMS. And interestingly, when you look at total score, the average total score is pretty much the same across all three syndromes. And it's not until we really break it down into these domains and modalities that we start to see these distinct phenotypes. We also looked at the relationship between the ABC. So here, the aberrant behavior checklist is one of our behavioral measures there's 5 different domains that are captured on the ABC. The R represents the value of the correlation. Anything shown here was the statistically significant correlation. And what you'll see is that we see significant correlations between overall scores on the SAND, as well as sensory seeking and hyporeactivity on both the SAND and short sensory profile for irritability. We see that the greater number of sensory symptoms, the more socially withdrawn and the greater levels of stereotypy were reported. And we see that hyperactivity was correlated on both measures with hyporeactivity. So this makes us really interested in looking more at attention. And then finally, looking at visual evoked potentials and our auditory steady state EEG responses and eye tracking, my main takeaway is that we're starting to be able to differentiate ADNP syndrome from other groups. So on the VEP, we see weaker amplitudes, so a smaller neural response compared to both controls and idiopathic ASD. When we look at the EEG response, we're seeing less coherence compared to idiopathic ASD. And when we look at eye tracking, on what we call a gap overlap task, where a stimulus is presented and then either another stimulus is presented overlapping with the initial stimulus, or there's a gap and then another stimulus is presented, we see in both of those conditions, we see that the latency of the saccade, so that looking at visual attention is delayed. And this work on EEG and eye tracking is coming out of Jen Fospike's lab. And then the last piece of data that I want to share is now trying to combine these two, trying to combine what we're seeing behaviorally with our EEG responses. And this is really interesting. So on EEG, this is a habituation task where a sound is played 4 times, and the initial time you hear it, as you'll see in controls, there's a large response, and then an individual habituates to the response as they continue hearing it. So overall, there was a correlation between the amplitude of the response and seeking scores. But when you really the way that we were really able to see the difference was to separate the group between the high seekers and the low seekers. And there you saw that the low seekers looked similar to controls, whereas the high sensory seekers looked quite different. So taking this all together, in terms of future directions, we are really interested in using sensory reactivity measures to look at treatment effects in the sensory domain, both in the context of clinical trials and behavioral interventions, and hopefully clinically as well, assessing the natural history of sensory symptoms in ADNP syndrome over time, so looking longitudinally, developing ADNP-specific norms to enhance our clinical outcome assessments and give us another marker of change. And then translational research through preclinical collaborations is an important step as well. And a huge thank you to all of you on this call, to our many wonderful ADNP families, to Christina Layton, my research coordinator, and our whole team that has made all this work possible.
Thank you, Paige. So we have a couple of questions and a couple of minutes for them. So one of them is, can a patient be hypo and hyper? Can you be?
Yes. So very good question, and the answer is yes. And I think this is where things can get confusing for people who aren't aware of that, right? So an individual can be. I just did a SAND where the individual was reported to seek out the sound of sirens, but was unresponsive to sounds such as the doorbell ringing, something dropping on the floor. We also see one of the interesting ones that has come up a couple of times for individuals who have a high threshold for pain and temperature is report of those individuals not liking the texture of hot or warm foods, for example. So we certainly do see within a given modality, within a given domain, we do see that we can see very specific sensory preferences within individuals.
Thank you. And then let's see. Can a child have ADNP and not be diagnosed with autism? Is that a misclassification? Is that an error? How do we understand these syndromes that have differing presentations?
Sure. So my general sense, and Alex can certainly chime in, is that autism is a spectrum, and that's exactly what we see in ADNP syndrome. We have kids and adults who are surpassing that threshold to very clearly meet DSM-5 criteria. And then we have other individuals who show certain symptoms of autism, but have other aspects of their functioning to see what we typically see in autism, and therefore don't meet the clinical threshold.
Have you looked at taste?
Have I looked at taste? No, we have not looked at taste. You know, it's really interesting. I think one of the challenges is figuring out how to measure certain other aspects of sensory reactivity outside of vision, hearing, and touch in a way where an individual does not have to give a verbal response. And so it's certainly something of interest, but we have not looked at it.
And there's a question here, which is a good one, about CVI. So visual impairments, and how does that play out here?
It's a good question. So this is something that we certainly account for, and we document very well the various visual and auditory impairments that parents are reporting. I think one of the benefits of clinical trials is that we're really looking, for the most part, at an individual's change throughout the trial, in which case the presence of CVI, as long as we can actually get the data, is less relevant because we still can see change. In the context of looking at group level data, that's sometimes where we pull out cases where there's very clear, well-known visual or auditory impairment.
Perfect. Thank you so much. I think we've gotten all the questions answered. Sandra asking specifically about reactivity to taste, but I think the short answer is we haven't looked at that, and someday. Thank you, Paige. So our next speaker is Alex Glebzon, who is, well, you all know him. He's a professor in the department. He is the head of the Child and Adolescent Psychiatry Division. He's the clinical director of the Seaver Center for decades and decades and decades. He was born at Mount Sinai. He never left Mount Sinai. I'm sure you all know what he's going to talk about, some of the clinical trial work he's been doing. So Alex, take it away.
Thanks, Joe. Good to be here. Saw lots of familiar names on the participant screen. I'm going to share my slides. All right, everyone can see the full screen. All right, so I'm thrilled to present some preliminary data, most specifically from our ketamine trial, but I'll talk to you just a little bit about our approach, what we were thinking kind of leading up to this trial, and then what we're thinking about going forward. So I don't need to kind of review this too much, but just to kind of orient everybody again. And I think Joe said, can I explain something simply? He said, no, I'm going to do my best to explain it simply. So as you know, ADNP is a gene on chromosome 20. It encodes for the activity-dependent neuroprotective protein. It acts as a transcription factor. It plays a critical role in regulating other genes and in brain formation and development. The syndrome is also associated with a lot of behavioral issues, and these are important because these are some of our targets in clinical trials. Move this for a second. Yeah. But things like autism, and then within autism, restricted and repetitive behaviors, sensory symptoms, as Paige mentioned. We see kids with a lot of attentional deficit, a lot of hyperactivity, impulsivity, and then there's a whole host of other medical features that are, of course, more challenging to target in clinical trials, at least short-term trials. So thinking specifically about ketamine, which is a glutamatergic agent, this is a study by Alana Gozes' group. I think Alana was on the call at one point. So ADNP syndrome has been linked, at least in some studies, to glutamate dysregulation. And glutamate is an especially critical neurochemical because it's the main excitatory neurochemical in the brain. It's responsible for neuroplasticity. It plays a huge role in learning and in memory. And so you see in this study that in mice missing, in mice missing one copy of their ADNP genes, this is replicating the human condition. There are actually deficiencies in the glutamate transporter gene. And that's in comparison to mice that are having 2 copies, sort of typical or wild type mice, versus mice that are being treated with NAP, which is a snippet of the ADNP gene. So the idea is that there's glutamate dysregulation. It's kind of localized to some extent in the hippocampus and the cortex. The hippocampus is, of course, the kind of memory center of the brain. And so this is giving you a little bit of evidence that this is an important chemical. We know it's an important chemical broadly, and we know that it's dysregulated in ADNP. So jumping forward a little bit, this is, I think, 2019, where Matt Might, who runs this artificial intelligence group at the University of Alabama, along with everybody's favorite ADNP parents, Matt Davis and Sandra Simoni, talked about the idea of how do we figure out a drug that potentially upregulates the expression of the ADNP gene. And so ketamine kind of popped up when this query was posted into the artificial intelligence machine. And it popped up for a number of different reasons, but one of the ideas is that we know ketamine is an NMDA antagonist, so it enhances excitatory neurotransmission, and it does that basically by sort of blocking or taking your foot kind of off the brake. So it's blocking an inhibitory nerve cell, which are called GABAergic interneurons. So here you see kind of a cartoon that I stole from a magazine. This is like a really, I thought, pretty good explanation of this. So here you've got the presynaptic neuron, the postsynaptic neuron. This is called the synapse. This is how sort of, chemicals sort of cross, and this is how the brain communicates with itself. And so ketamine blocks an NMDA receptor on what's called a GABAergic interneuron, sort of releasing the brake and then leading to kind of enhanced glutamate neurotransmission. So there's an upregulation or increase in glutamate. And glutamate, as I said, is kind of an important excitatory neurotransmitter, and it's promoting synaptic plasticity. It seems that also ketamine promotes nerve cell growth factors, and so again, to kind of influence synaptic plasticity. So ketamine can be sort of a scary medicine. It gets a little bit of a bad rap as a party drug. If you use way too much ketamine, you can get brain damage. It's certainly toxic in animal models, certainly at high doses. But there seems to be some evidence now that if you use low dose ketamine, A, it's quite safe, but B, it can actually block the effects of the toxic effects of higher doses. And that data is sort of coming out of a number of different studies. The first one shown here on the top suggests that the silencing, essentially, of the ADNP gene can increase cancer growth in animal models. And then ketamine actually increases ADNP expression and then reduces cancer growth. That was sort of the finding from that one study. And these are some of the main studies that kind of led to this notion that ketamine could be an important compound in ADNP syndrome. The second study shows that low doses of ketamine before full sedative doses actually can prevent the toxic effects in cortical nerve cells. And then in the third study, what you see is pre-treatment with low dose ketamine, and actually low dose in this case is not so low dose. This study, I think, used 5 milligrams per kilo, which is actually a pretty high dose. It's a highly sedative dose, that's for sure. But that so-called low dose reduced cell death in the ketamine-treated rats when high doses were used. This third study also suggested that ketamine can enhance the expression of ADNP in cortical tissue. So sort of on the basis of all these things, Sandra, Matt, they came to us, they said, we think this is a good idea. This was not my idea, this was their discovery. After a little bit of convincing, we thought, yeah, this sounds like a great idea. So we set about designing a clinical trial. The idea was just to give one single infusion, really focused on safety and tolerability. So we selected 10 kids. We gave them all 0.5 milligram per kilo. This is a relatively standard dose based on studies in depression in adults. And then we monitor kids a day after the infusion, a week, 2 weeks, and 4 weeks later. We were interested in, again, safety and tolerability, but we were also interested in outcomes around behavioral symptoms, and particularly the behavioral symptoms that I mentioned earlier, things like social functioning, restricted and repetitive behaviors, ADHD symptoms, and sensory symptoms. So, right, I guess I mentioned this briefly. So again, safety and tolerability was our primary aim, efficacy was our secondary aim. Third, we were really interested, and we continue to be in this idea of electrophysiological markers and computerized eye tracking. I'll talk about that a little bit, but we are really challenged in terms of our clinical outcome measures and trying to find measures that are more objective. So we're constantly trying to pilot and assess the feasibility of more objective measures in the context of clinical trials. I think they're also critical potentially going forward in terms of trying to predict treatment response in advance. And then again, because of preclinical data suggesting that that ketamine can increase the expression of the ADNP gene, we were interested in using blood-based biomarkers with RNA sequencing to specifically measure ADNP expression, and also DNA methylation analyses, which we know are relevant to ADNP syndrome. So in terms of our results, as I said, 10 kids were enrolled. 3 of them were female, 7 of them were male. Age was about 9.5 years old on average. The enrollment criteria were 5 to 12. All these kids were intellectually disabled. Only to the question before, only about 4 met research criteria for autism spectrum disorder, and 7 met criteria for ADHD. 6 had frame shift mutations, 4 had nonsense mutations, and 2 identified as Hispanic or Latino, and the rest were white. So most importantly, safety. I think we were pretty confident that this was a safe drug. During the infusions, there were actually no adverse events at all, except for 1 patient who had some nausea and vomiting. And this is a participant who had a long history of pretty frequent vomiting. So it's not clear how much was related to the drug. But there were no clinically significant abnormalities in laboratory or cardiac monitoring. And that's one of the kind of important risk factors for ketamine, is it can really significantly increase heart rate and blood pressure. That did not happen. There were no serious adverse events. Some of the most common side effects that occurred over the course of the 4 weeks that we were monitoring kids included things like fatigue, some increased aggression. Some of the kids got more outgoing or elated or silly. There are some parents that reported a decreased appetite, increased anxiety, some restlessness. So these are all side effects that occurred in the context of the trial that represented a change in frequency from baseline, but these aren't things that we can necessarily attribute to the drug. And again, they were all occurring a day, a week, or even up to 4 weeks after. We just have to document everything for the sake of being as inclusive and careful as possible. But we definitely felt that the conclusion was that this was safe, at least at this dose, and in a single dose, of course. And then the exciting stuff, the efficacy. Now, again, it's really important to understand that this was a very small trial. This was a single dose, and this was what's called an open label study, meaning there was no placebo control. So I knew, and the patients knew, and I guess the kids knew, everyone knew they were getting medicine. And so there was a lot of optimism, there was a lot of enthusiasm, and there was a lot of kind of hope infused in this study. And so it's not unusual for these kinds of open label studies to have very positive findings. And of course, we had very positive findings. But what's important is that we were using measures that were collecting information from parents as the caregivers, collecting information from clinicians, and then we also had some more objective measures. And there seemed to be a lot of convergence, particularly around the symptom domains that we were hoping to see change. So autism symptoms, so social withdrawal, social avoidance, social communication, the restricted repetitive behavior, sort of the obsessive compulsive kinds of behaviors. We also saw improvements in ADHD symptoms and in sensory seeking. So I'll go through a little bit of that right now. So we use this measure called the aberrant behavior, the aberrant behavior checklist to measure social withdrawal. The anxiety, depression, and mood scale has a social avoidance subscale. And then we used a clinician tool called the visual analog scale, where we queried specifically around issues of social communication. And we saw significant improvement between baseline and after 1 week. We focused mostly on that sort of post 1 week assessment, but we did see significant improvement compared to baseline in this domain. Likewise, in the obsessive compulsive domain, as measured by the ADAMS, the repetitive behavior scale, and the visual analog scale, we saw again, pretty significant change over the course of that first week. And same with hyperactivity, inattention, using the ABC, the ADAMS, and the Conners. These are all pretty standardized measures of these types of symptoms. They have not been validated specifically in ADNP syndrome, but we do think that they meaningfully capture the phenotype. And so this was all kind of very, very promising. And then to what you heard this morning from Paige, using the short sensory profile and her SAND assessment, we also saw improvement in sensory symptoms. What's important here is that when you see the increase in scores on the SSP, that actually reflects improvement. So the higher scores are more towards typical development, whereas the SAND decreased scores reflects improvement. And so we saw improvement really across both these domains, both the sensory domain across both these measures, one of which of course includes an observation as Paige presented. So this is all, very, very promising and certainly got us enthusiastic and led us to start thinking very carefully about next steps. So in terms of next steps, I think, we have a lot of challenges in terms of really being clinical trial ready and making sure that the next study is designed in a way that maximizes our chances for detecting, improvement if improvement is to be found. According to the FDA, and that's one of the important things here, treatment benefits determined by how a patient survives, feels, or functions as a result of treatment. So the question is, how do you measure that? What kinds of clinical outcome assessments can you use? So clinical trials will often use patient reports. They'll ask a patient, how do you feel? How are you functioning? And that's not something we can do in ADNP syndrome. So we rely a lot on caregiver reports. Caregiver reports, as you know, are they're subject to a lot of bias because caregivers, want their kids to get better, and they're coming into our center and they're optimistic and they're enthusiastic, and we're optimistic and we're enthusiastic. So we're all kind of subject to bias. Same with the clinician reported tools. There's been some work in other neurodevelopmental disorders to develop kind of more composite measures where you're combining tools across different domains. And then most importantly, the idea of trying to develop more objective tests that are totally blind to treatment condition. Doesn't matter, an MRI scanner, an eye tracker, doesn't matter. You'll see through any kind of placebo effects. So one example of a tool that we're working to develop in ADNP syndrome that's already been adapted for Angelman syndrome, for Rett syndrome. It's being adapted for Phelan-McDermid syndrome. These are all kind of single gene disorders associated with autism that have the same challenges that we have in ADNP syndrome. But the goal is to take a very standard measure, what's called the Clinical Global Impression Scale, and adapt it and anchor it specifically to symptoms of ADNP syndrome. So here in this study, we reviewed the literature, we interviewed caregivers. We've actually done that already, probably with some of you, in ADNP syndrome, to try to figure out what are the most impactful symptoms to parents. That's a really kind of important metric from the FDA's perspective. We also want to engage expert panels, so clinicians, to really operationalize the symptom domains that are identified. We have to develop very kind of concrete and clear rating anchors so that when we say something is improved, multiple raters would agree that it in fact improved. We have to figure out how much improvement would be considered clinically meaningful. And then there was a lot of work done to generate sort of mock patient vignettes and then test reliability and standardized rating trainings across the various raters. So this is work that's just begun now in ADNP syndrome, and we hope to at least have some solid tool available to pilot in the context of the next clinical trial. Paige mentioned her work with this very exciting tool, the sensory assessment for neurodevelopmental disorders. This is really the first clinician observation and corresponding caregiver interview. It's really meant to tap into these sensory reactivity symptoms. There's now, as Paige mentioned, lots of data to support its use, including in ADNP syndrome. I think as you heard already this afternoon, children with ADNP syndrome show significantly more tactile hyporeactivity and sensory seeking behavior. We've already used this measure, as I said, in the ketamine trial. So not only is it a good kind of phenotyping tool, but it also appears to be sensitive to change over time in the context of treatment. So that's a really kind of important metric as far as tools are concerned. So we certainly intend to continue using this tool, and it's got this kind of more objective component to it. Paige also mentioned computerized eye tracking, and so we use that in the context of the trial. We did computerized eye tracking at every visit, actually. And so two main tasks were used, both measure attention, one sort of a visual attention to social stimuli. It's called the gap overlap task. So it measures, I think as Paige mentioned, the engagement and the disengagement of visual attention to both social and non-social stimuli. So you can actually also measure preferential social attention. And then the joint attention task, which is actually, it's measuring the ability to kind of follow the eye gaze of a character on the screen as they kind of look at either the target or a distractor. So this kind of joint attention is sort of a very, very critical component of social attention, basically. And the results from this tool in the context of our trial suggest that actually both have some promise and certainly some feasibility in the context of clinical trials. We saw change in a number of these measures that are very, very promising, actually. So we'll also continue to use this and pilot this in future trials. And this is work that was led by Jen Foss-5, which is why her cool picture's up there on the screen. Jen's also working a lot on EEG biomarkers. This is when your kids get hooked up with those huge nets with 128 leads. We focused on this study in something called the auditory steady state response. This is where participants will hear a series of clicks at a stimulation rate of 40 hertz. the auditory steady state response is recorded with these EEG nets. And it's really where the coherence between the trials of clicks is measured with a time frequency analysis, and it reflects the synchrony or the integration between nerve cells. ASSR is considered a measure of the balance between excitatory and inhibitory or glutamatergic and GABAergic neural systems. So we think it's obviously it's an important area. It's particularly relevant for ADNP syndrome. ASSR actually might be correlated with sensory symptoms, and we did see significantly increased intertrial coherence over the first week of the trial in the context of ketamine treatment. So that's what you see here with this increased intertrial coherence. So these are all, again, very early stages, very early pilot studies, but extremely exciting to be able to show EEG changes in the context of ketamine treatment. So to summarize, ADNP syndrome offers a unique opportunity to study a specific genetic cause of ASD. And Joe mentioned this. I think it's really, the idea that the whole autism world is excited about rare causes of autism, like ADNP syndrome, is really important because that's going to lead and generate more funding for ADNP in particular. But the idea that you've got a more homogeneous neuropathology that can be targeted for treatment and then apply it to autism more broadly is really very exciting, and it convinces people, pharma, NIH, whomever, that there's a really kind of broad relevance to ADNP syndrome. We still, though, need to do a lot of work to carefully phenotype. We probably need to do some good natural history studies. We have to be very thoughtful about which targets we want to select for clinical trials. We definitely need better clinical outcome assessments. Some of those are ongoing, some of those have been done and will continue to be piloted. We need to do more to develop and validate our biomarkers. We've taken some really good kind of first steps, but there's a lot more work to do. But as I said, I think it's really very exciting that eventually treatments in ADNP syndrome will be used to sort of parse autism more broadly and predict who might respond to treatments within autism based on response in ADNP syndrome, and kind of shared underlying pathways or shared phenotypic profiles. So all really very exciting. So next steps. So I think certainly these data, while very preliminary and of course open label, are enough to suggest that it's worthy of further study. So we intend to design a placebo-controlled trial and a treatment trial, so beyond just a single dose. There's a lot of questions about delivery. Ketamine can be given intranasally, subcutaneously, intramuscularly. It can be given orally. It can be given IV. So we think that probably the best data in terms of the bioavailability is around intramuscular administration. So we will likely switch to an IM administration for better ease of use. The treatment will be, instead of one single dose, it'll be 8 doses. We're thinking probably over about 4 weeks, and we'll use a crossover design. So that's depicted here, where people receive either placebo, see my mouse? They'll receive either placebo or ketamine for 4 weeks. There'll be a 4-week washout period, and then they'll flip to the other condition essentially. That way every patient can be kind of their own control. Every patient will get access to drug over the course of the trial, and it also allows us to keep our sample size a little bit smaller. We've already submitted to the FDA a new drug application. We actually have to now revise it based on comments from the FDA. And we intend to select additional sites so that we're not burdening families in terms of travel. So we'll try to get sites that are relatively well distributed and make sure we get access to as many patients as we possibly can. And the last thing we have to do, obviously, is get some funding. So that is all that I have to present. Thank you so much for our group, everyone that worked so hard on this study. Thank you so much to the families, to Sandra, to Matt. And I guess I should have mentioned it earlier. This was probably the fastest funded and fastest completed study I've done in my many-year-long career in clinical trials. So it was really very exciting. This is probably one of the most exciting scientific stories I've been working on since I started this. And we're really looking forward to the next steps.
Thank you, Alex, for a great talk. And we have some time for questions. So I think people are questioning about, exclusion, things like that. Somebody, for example, are there any drugs that would make a child not eligible to participate in the next trial? And I know it's probably not. I mean, there's a few questions like this. We'll take a few, but we probably won't be able to take them all.
Yeah, so I think that we will likely design the trial around roughly the same ages. It's important in clinical trials to keep the age range relatively narrow. We will likely keep them to sort of pre-pubertal kids around 5 to 12. There's some flexibility there, but but I think that sounds like a reasonable first start. In terms of medicines, there are not any medicines that I'm clear about that would necessarily be in conflict, but we will be asking families to keep on stable medication regimens for probably about 3 months before the trial, and of course throughout the trial as well.
Right. And so I think it's important that the next trial is going to be a trial. It's not going to be. It's hard to get data if it's a wide open trial for every age group. So that's just the reality. But one question is about any evidence that ketamine would be more effective on younger versus older kids. And I think, go ahead.
Yeah, that's a great question. I think we tend to think about brains as more plastic as they're younger, and we tend to think about younger children as kind of having more room to grow. But we also have to balance that against some safety and also about the relevance of our measures. So I think trying to capture younger kids, but not going quite as young as, like, let's say 2, at least as the first pass, I think it's the right place to start.
And of course, there's a lot of questions about international trials, international sites. If you're going to do multi-site, is it going to go international right away, or is that going to be the next phase?
Yeah, so we've been approached by a number of different sites. I think we are generally open to sharing as much as possible our protocols. If it becomes partly a question of funding, but it also becomes a question of design. I think the most important thing for the next study is to have enough patients to really feel confident in the efficacy of the medicine and to be able to put out a really convincing paper that says this drug works and it beats placebo. And you need a certain number of families to do that, and if certainly we would need multiple sites in the U.S. in order to make sure that patients aren't constantly having to travel. And if there's sites outside of the U.S. that want to participate and have funding to do so, we were thrilled to share and sort of combine data after the fact, likely.
And since you mentioned infusion and IM, so maybe you can just go over that again. What are the benefits to the different approaches? You mentioned, I think, the IM.
Yeah, so when we do the infusions, this is an intravenous infusion. It has to occur in a clinical research unit. It has to occur obviously on site. You need to have very close monitoring by an anesthesiologist. The infusion is quite slow. It's usually over about 40 minutes. If you infuse a 0.5 milligram per kilo of ketamine immediately, you could basically sedate somebody. Whereas intramuscular, it distributes a little bit more slowly. It's still highly bioavailable, I think up to like 93%. I think it's much easier. And if there's any hope potentially of being able to train parents to do it at home, intramuscular injections are actually pretty simple to administer once you get used to it. And IV is impossible to do at home.
Yeah. And then there are a couple of questions about longer term results at week 4 or later.
Yeah, so we didn't present those, but in fact, the results were maintained for the most part. Numerically, there was a little bit of a reduction in improvement, but those improvements that you saw at week 1 were maintained up until week 4 for the most part. We didn't exactly know what to predict and how long out the benefit would extend for after only a week, after only a single infusion. I think if you look at depression studies, for example, after a single infusion of ketamine, there is evidence that depression can continue to improve over the course of several weeks. But we did see a maintenance of improvement even up to week 4.
And then, if funding aside, what's the timeline? Like the FDA, you've already gotten one response back from the FDA. You have to make some modifications to protocols. You've talked about endpoints. You've talked about many things, and inquiring minds want to know. Yeah,
I think so. Funding aside, right? So I think if over the next couple of weeks we can finalize the revision to the protocol and submit it to the FDA will probably take about a month to review it. At that point, we would submit to like regulatory authorities here, start to find other sites. I mean, at the most optimistic, it would be 6 months, frankly, before we could start, assuming that the funding was in place.
Yeah. All right, that's nothing wrong with being optimistic. And then if ketamine does work, so is that a forever treatment? Do people have to take it for life? What do you think is going to happen there?
Yeah, so that's a really good question also. I mean, I don't think ketamine is the kind of medicine that you want to be taking every day for the rest of your life. I think we would have to do a lot more in terms of what the long-term safety looks like. So when you think about like depression, for example, and esketamine, it's essentially given for 8 weeks or so, for 4 weeks, I think, twice a week. I'm not sure exactly what the protocol is. And it's given in sort of boosters over the long term. I think probably we'd have to design something similar to that. It wouldn't be like a chronic everyday treatment. And it would just be about balancing the benefits versus the risks.
Thank you. I think there's some specific questions about how much funding is needed, but I think we have to probably take that offline because it's multi-site. There's lots of things that go into it.
Yeah, so we're talking with Sandra and the foundation. I know that just to say, right, the funding from the original trial was raised within a month from the foundation and the families. And so we have a rough budget, and I think we'd be happy to share that with you, and Sandra would be happy to share that with you, because I'm sure that families and the foundation will be called on to help fund this. We did submit a grant also, so there's a lot of different opportunities here to get funded.
Yeah, and then people, of course, are aware that there was blood taken. So anything come out of the blood work? So
yeah, that's another great question. So those analyses are ongoing. I think the first pass was not quite as optimistic as we were. It was not as positive as we hoped it would be, but in terms of the RNA sequencing data. But I think that story is sort of unfolding as we speak. So I think I purposely did not present it because I want to make sure that we have the right story before it's really put out there.
Perfect. Well, thank you so much. I think we've even caught up on the questions, more or less.
Great.
Thank you, Alex. And so our next speaker is Nan Yang. Nan Yang is an assistant professor in neuroscience, and she has become a close member of the Seaver team and has been developing these iPSC or pluripotent stem cell models for ADNP syndrome. And as I mentioned, with these kinds of cells, you can not only understand what ADNP does, but you can also possibly test drugs or even discover drugs. So Nan, thank you for presenting, and take it away.
Thank you, Joe, for the introduction, and good afternoon, everyone. Thank you again for spending your afternoon with us and let us share our work on ADNP. So my group, as Joe briefly mentioned, uses human stem cell-based models to study the development and function of the human brain. Together with the Seaver Center, we are building human model systems to investigate the function of several genes associated with autism, including the ADNP syndrome. So in today's talk, I will first give you a very brief introduction to human-induced pluripotent stem cells, or the iPS cells, including what they are, why we use them to study human psychiatric disorders, and how we use them. I will then talk about some of the current work from our group that uses the stem cells or iPS cells derived from patients with ADNP mutations and their unaffected family members to try to understand this disease. So to understand what iPS cells are, we have to take a slight detour and go back to the developing human embryo. The human embryos reach the blastocyst stage for 5 days after fertilization, at which time they consist of 50 to 150 cells that can grow into different types of cells in the body, including the cells in our brain, as well as the cells that goes to the germline. Their potential to generate all other cell types of different germ layer is described as pluripotency. In 1998, James Thompson successfully isolated the inner cell mass from human blastocysts and maintained them in cell culture. These cells exhibit a remarkable capacity of long-term proliferation. At the same time, they can differentiate into 3 germ layer cells, including the ectoderm, mesoderm, and endoderm. That gives rise, the ectoderm gives rise to the brain cells. These cells are named embryonic stem cells. Their potential to be used in human developmental biology studies, potential drug discoveries, and even transplantation medicine was immediately recognized. However, you can also imagine the difficulty in deriving these human ES cells. ethically and technically. Therefore, it is probably not surprising to tell you that one of the ESL lines that we are using in the lab, even today, was derived in this very first study. Actually, it's one of the cells showing in this picture. So wouldn't it be nice if we could generate something similar to the human ESLs that can be maintained in culture infinitely, but still possess the ability to differentiate into different cell types? And the short answer is yes. In 1962, the scientist John Gurdon removed the nucleus of a fertilized egg from a frog and replaced it with the nucleus of a cell taken from a tadpole's intestine. And this modified egg grew into a new frog, proving that the mature cell can return to an immature state and form all types of cells. However, do we need everything from a fertilized egg to reprogram the nucleus of a fully mature cell back to its pluripotency? The answer is no. In 2006, Shinya Yamanaka successfully identified 4 genes, when activated, can turn the skin cells back to its pluripotency. And this marks the birth of the induced pluripotent stem cells. Both of them shared the Nobel Prize in Physiology or Medicine in 2012 because of their discovery in pluripotent reprogramming. We are very excited about the iPSL technology because now we can derive primary cells from the patients, including fibroblasts or blood cells, and reprogram into iPS cells. iPS cells can then be differentiated into different types of cells within the body, including the neural cells, which is the center of our study. But why is this so important to have these human cellular models? What are their advantages over model organisms, including the broadly used mouse models? Well, there are several reasons. First, because the mouse brain and human brain are very different. Not just in their sizes. If we scale the mouse brain up, this is a mouse brain, you can see it has a very smooth surface compared to that of the human brain. These characteristic folds of the human cerebral cortex greatly expand our brain surface. Underlying the structural changes are differences in cell composition, proliferation, migration, and differentiation. Therefore, many cellular features unique to the human brain cannot be modeled or studied in the mouse. In addition, there are many genes that play very different roles in mice and human. But more importantly, or more related to the disease research, is that humans and mice carry very different genetic information. For example, many disease-associated genetic variants cannot be identified in mice. Additionally, I think I saw one of the questions in the chat that why different people carrying the same mutations can have different disease manifestation. This is at least, we don't fully understand the reasons, but this is at least partially due to the genetic modifiers in individuals that will be even more difficult, if not impossible, to model in mice. And we hope to find at least some of the answers by using a human system that is more physically relevant to the disease condition. Then the next question is, what type of cells and cellular processes we want to create using the human iPS cells? The large-scale genetic studies led by Dr. Buxbaum, among others, give us some guidance. They found that the expression of autism risk genes is enriched in the excitatory and inhibitory neuronal lineages. So neurons in our brain do not work independently. Instead, they form networks through the tiny structures that are called synapses. At the synapses, neurons can talk to each other by secreting neurotransmitters. Excitatory neurons can secrete glutamate that can make other neurons more likely to be active. In contrast, inhibitory neurons using something called GABA to make others less likely to become active. excitation and inhibition are the two major modes in the brain circuits. When the balance between E and I is disrupted, many disease conditions may occur, including autism spectral disorders. So during the past few years, we have developed methods to generate the excitatory neurons and inhibitory neurons from human iPS cells. We establish neural network using such neurons in a petri dish, and the cells may or may not carry any disease-relevant mutations. And we use many different approaches to investigate the possible pathogenic changes associated with different mutations. Synaptic transmissions could be measured, which is just how the neurons talk to each other can be measured experimentally to uncover the functional abnormalities. The most sensitive and quantitative approach is called patch clamp. And here is the sample trace of currents that we recorded. So this is an electrode. We use this to record responses directly from the individual neurons. And this is one of the sample trace of the currents we recorded. And here is what we find in the control cells that do not have any mutations. And the bottom one is after we use something called shRNA to reduce the expression of a disease-relevant genes. And you see just by vision, you can already see much fewer peaks, which means much less frequency of firing. However, this method is very low throughput. Just to give you an idea, we need to patch individual cells using electrode connected to an amplifier to record the currents, and this may take a few minutes. So to get a better idea or a faster idea about whether one mutation can cause any differences at the synaptic level, we also use a method called multi-electrode array. So this method, when we plate the neurons in the wells, the activity across an entire population of cells can be detected. Using our current setup, we can measure the activities of neurons in 48 wells at the same time. And this method may not replace the patch clamp because of the resolution is not as great, but it can provide evidence if the neuron property or synaptic transmission is impaired in the mutant neurons. And this is extremely important because, again, we have many, many different mutations detected in ADNP gene that can all be pathogenic. In the next few slides, I will share with you some of the work led by a former postdoc, Gizem Inagerbach, from our group on ADNP. So this diagram summarizes some different pathogenic mutations found in the ADNP gene. And the Seaver Center has collected the blood cells from many families to generate iPS cells. The first question we ask is if any of the phenotypes can be explained because of the changes in protein localization caused by ADNP mutations. And the reason is ADNP executes its function mainly in the nucleus. To do this, Gizem focused on three very representative mutations. One is a recurrent mutation, and the second one happens basically in the very beginning of ADNP protein. And then the last mutation is here, which means it's almost in the end of the protein. So when she expressed different mutant ADNP genes and as well as the wild type ADNP protein, she noticed something very interesting. So she noticed that this is a wild type ADNP protein, and these red cells are the neurons. And you see a very round signal that overlaps with DAPI, meaning the ADNP enters the nucleus. And she observes the similar phenotype or the similar pattern when she checked the reoccurring mutation and also the other mutation that happens almost to the C terminal of the protein. However, she did not detect any protein expression when the ADNP has a mutation that is at its very end terminal. So using this method, we are trying to profile quickly where the mutations are and what mechanisms that we may have to look into to find how the mutations can change the function of neurons. And we're still carrying out these experiments and expand our assay to many different mutations found in ADNP gene. Gizem also measured the gene, Gizem also asked if there is any phenotype that she can detect when the neurons carry the recurring mutation, which is the tyrosine 719 to stop codon change using the human excitatory neurons. So she performed MEA, the multi-electrode assay, using the excitatory neurons generated from the control cells and the mutant cells. And she immediately noticed the two differences. The first one is the number of active electrodes, meaning the electrodes that detect any activity is different. And there is much fewer when we have when the neurons carry a mutation. And she also noticed a reduced mean firing rate and number of bursts in the mutant condition. So putting this together, it suggests that in the excitatory neurons, at least, when the neurons carry this mutation, there is a reduced network activity. Gizem also measured the gene expression in the mutant inhibitory neurons carrying the same mutation. And this time, she uses iPS cells derived from two different families, differentiate them into inhibitory neurons, and performed RNA sequencing to profile the whole transcriptome. This experiment identified 624 differentially expressed genes, which means they can be either upregulated or downregulated in the mutant neurons when you compare them to our control cells. And more importantly and intriguingly to us is that the genes whose expression levels modulated by the mutation are significantly involved in biological pathways, including chemical synaptic transmission and nervous system development. Together, this data suggested that there are changes in synapses when the neurons carry the recurrent mutation as listed here. So we have started performing these experiments almost 2 years ago, but it's really interrupted by pandemic. Now with all our preliminary data, we're searching for more funds to expand our research and what are we going to do next? So first, we want to perform more MEA experiments using all the patient-derived lines that we have collected, and so that we can characterize the cellular and molecular mechanisms underlying the pathophysiology. We want to identify or hope to identify robust and reproducible phenotypes so we can ultimately use them to look for either pharmaceutical or genetic manipulations, as some people asked or discussed in the chat, such as CRISPR, to reverse the phenotypes observed in the patient-derived neurons, which will place us ultimately at the position for drug discovery. And in today's talk, I shared some of our work that uses human stem cells to study autism. I sometimes get questions from parents about stem cell-based clinical trials. Well, I am not a clinician and cannot provide professional advice. But before I end my talk, I want to introduce the resources provided by the International Society of Stem Cell Research. So here you can find resources on many important topics, including what you may want to learn when you consider participating in such clinical trials. And I hope this will be helpful to everyone. And with this, I want to thank you for your attention and your interest in ADNP syndrome, as well as for your support for our research. And here are the people without whom none of the exciting work can be done. Thank you again.
Thank you, Nan, for a great talk. So I'm gonna add a little bit to your presentation, if you don't mind. I think because most of the people, many of the people who are listening, they know Alex, they know Paige, they know me, they know Anna. But I want to just take a step back because this is fairly complicated stuff. It's kind of a lot of science, but I think it is one of the most remarkable breakthroughs in probably all of science at all times. Yamanaka won the Nobel Prize 6 years after the paper came out, which is one of the quickest Nobel Prizes ever awarded. And the reason it was awarded is because suddenly you could study tissue from a human being without actually doing anything, hurting the human being. So we could study brain tissue without actually taking somebody's brain, which turns out to be a pretty nice thing to not have to do, right? And so even though it's kind of a big mouthful, iPSCs, induced pluripotent stem cells, all those sorts of things, it is absolutely a major focus of every group in the world, any academic or industry group that's thinking about human disease, because you can grow, in our case, brain cells in a dish, right, without actually sampling somebody's brain. And so Yamanaka showed that you could reprogram some cells, and he, for example, showed that you could reprogram heart cells, right? But a lot of the work to make neurons, to make nerve cells, happen in other groups, and Nan actually is one of the leaders of both of the methods that are most widespread in how to take a blood sample or a skin sample and turn it into a subtype of a brain cell. So it is not only a remarkable technology and completely, changes the game for everybody in any kind of disorder of the human body, but also Nan has been a leader in this, and we're very happy that we could twist her arm to join the ADNP team. So I don't see any specific questions for you, and I think it's because there's something very technical about reprogramming blood into brains and then doing electrophysiology. But it is, I think, very important and interesting that we have this resource now, and that we can now do a lot of the work that you've mentioned.
Thanks.
And maybe actually, so here's a question which I think we should address that just showed up. Is this stem cell technology for research, or is it also for clinical treatment?
So, so far, we're most confident to use this for research. And there is something called regenerative medicine, meaning we do want to develop someday into a therapeutic method, but we are far from that. That's why in my second last slide, I'm trying to share with you the resources from the International Stem Cell Society that gives you more prospect about how stem cells are these days being used. But again, we have to be more cautious about its translational applications at the moment, but that's a hope.
And I guess I would add to that the stem cell technology that you're using will be perfectly fine for discovering, for example, a small molecule or a gene therapy and testing it. So in other words, you can do all the preclinical work in cells and then do actual clinical treatments, not with the cells themselves, but what you learn from the cells.
Right, the cells actually have been used for even test the toxicity of drugs. That's a really good way to use them. And should I move on to the next question?
Maybe well, you can, if you want to. Take it away.
So I think I only talked about excitatory and inhibitory neurons in the current talk because these are the two most relevant cell types as identified or discovered using genetic studies. But at the same time, we are also generating the other different neuronal subtypes using different neurotransmitters, as mentioned in the question about, for example, noradrenaline, serotonergic neurons, or dopaminergic neurons, because they can all play a role in disease development. So the short answer is we're also looking into other cell types, but we're just more focused on the two major cell types that seems to be most relevant at the moment.
Yeah, and maybe to give a little bit of background, the questioner asked about these other kinds of neuromodulators that Nan just mentioned, and one of the reasons is because some interesting results with atomoxetine, which is an ADHD drug that impact some of these same pathways. So asking the question about whether we want to also look at those cells in the brain that work in serotonin and other pathways. And I am going to bring up a slide I was going to bring up later, but since, let's see what we're doing for time. Oh yeah, I'll bring it up now. Do you mind if I jump into your talk for a moment?
No, of course, no problem.
Very relevant. It's very relevant. One of the things that you know, is the right slide showing?
Yes, I think so.
One of the things that I've mentioned and it's been a major focus of Anna Kostic has been developing partnerships with both industry and other academic sites, and to no small degree, all of those collaborations are around these stem cells, right? And some of these are in early stages. There are other ones that are that we're still working out. But Rumi Scientific is a local company in New York City that's taking these stem cells and not just making neurons, but making something called an organoid, which is neurons that come together and form, what you might call a mini brain. And this company has developed a methodology for imaging using microscopy organoids very quickly and seeing if there's some disruption in the way the cells are supposed to be layered. So in your brain, cells have a very specific layer. And so we've been collaborating with them for over a year now, almost 2 years, and we just initiated a second collaboration to extend it because we think there might be something going on there. There might be some changes in the organoids with the ADNP mutations. And if there are, then it could be used to screen drugs. New Site is a company that also works a lot with stem cells. New Site actually is closely affiliated with Nan's previous position. And again, it's the same idea. See if they can see differences in their specific assays with the stem cells, with neurons in Petri dish. If they can, then see if they can't turn it into a platform for screening drugs, right? So the things that Nan was talking about, we also have reached out to companies that specialize in this. And each of these companies has a specific technique that they're expert at. So we're taking a broader view, or Nan's taking a broader view, looking at many things. But there are also companies that have, a specific test, and if it fits, if these cells show something in that test, we get to jump ahead. And then Tim Spicer at Scripps Institute is exactly the same thing. He's developed a very specific assay for making neurons in a specific way from stem cells. And with the same idea, we shared some of our stem cells with him as a collaboration, so we could see if there were differences with the idea of ultimately developing a screening protocol. As of now, we don't see in his system a specific ADNP change, but we're still examining it quite extensively. So those are things, as I said, that Anna Kostic is leading, but they're basically a follow-on from all the stem cell work that Nan has developed, and they're taking advantage of different groups or companies that have a very specific assay they've developed. And if ADNP, the cells fit in that assay, it's good for everybody. And if they don't, we just move on. So here's a question. I don't know if you want to try it, but how would you investigate a de novo variant of unknown significance in the ADNP gene? What functional studies would you recommend? Are patient-derived cells an option?
I think patient-derived cells are an option. And the other option is even doing gene engineering to put the variant into the cells. It will be more difficult if we don't know the significance. But the short answer is we can just test what we usually do with our patient-derived lines for the function, morphology, gene expression in the cells. So yes, we can look into the potential changes that may generate any pathological consequences.
Yeah, and there are also in silico and biochemical ways to look at variants of unknown significance. Somebody wants to email me a Voos, I can take a look at it and talk to some of our geneticists and functional people and see what we think. But the ADNP syndrome field is fairly young, right? It's not like, with breast cancer gene BRCA1, BRCA2, we pretty much can make a prediction with almost every mutation about whether it's deleterious, because a lot of work has gone into that. And ADNP isn't quite there yet, but it's certainly a goal. But I would say that clinical geneticists are take the most conservative line. They're supposed to, but we can look at any mutation and give an opinion about what could be done to validate it as whether it's functional. But again, it's kind of early days, and we don't yet know which functional assays are going to be most related to clinical phenotype, for example. Okay. And a lot of thank yous from the participants for all the great talks. And some of the things I think we can. It's going to be a group conversation right after, in a few minutes, and I think at that point we could talk about some of these larger open questions. So I would like to invite, again, thank you, Nan, and I'd like to invite Sandra Simone, who wants to talk about some of the new progress and new initiatives and all the great things that are going on in the foundation.
Hi, Joe. Thank you.
Hi.
I just want to say thank you, first of all, to the entire Seaver team. You guys are awesome, and our families are very interested in all of the science and research that's happening. So these days of learning sessions are very important. We hope they continue. I wanted to just quickly announce to our community that we have just received IRB approval for our patient registry. So this is very important to get IRB approval, because we can, it's important for the use of security of the data, and our researchers can use it moving forward. And this will be an international patient registry. So it's open to all individuals with ADNP syndrome around the world. We will have short-term and long-term information entered. So you will go in and create an account for your child and put in developmental information and genetic information. And then we have, as Paige was talking about, these validated surveys. We will have several validated surveys that you will continue to update every 6 months. So we will be creating information for a natural history study, which is one of the most important things we need. These surveys are validated through the FDA, so our researchers can use it. And this research information that we collect will be available to all researchers around the world. So right now, there currently exists very little data in one place. So Seavers might have 20 kids. There's a paper on 70. It's not all combined in one place. So this is very important to our community. We have about 380 patients that are in our group, and more out there probably. So getting everyone to participate in this is very important, because Joe and his team can use that information and learn more about ADNP very quickly. So that's all I wanted to update everybody on. Oh no, sorry, one more thing. We're also launching a biorepository, also beneficial for the Seaver team. We have created 5 IPC cell lines, and we're making 3 more. Those will be made available to Seavers and researchers who want to use them. And then we are going to open a blood biorepository at Coriel. Also will be available. It's through an NIH-funded platform. So we're going to be sending out all this information if you've signed up on the contact registry on our website. So I encourage you all to sign up on that, because the more samples we can get in our biorepository, the more information Joe and everyone like Nan can use if they want. So that's it for my update.
Well, thank you. And thank you for the update, but also thank you for everything you do. I know that the families greatly appreciate all your efforts, and we at the Seaver Center also appreciate your efforts, and we find it really is a wonderful relationship between the ADNP Foundation and the Seaver Center, and it really makes it fun to come to work, shall we say. So we're going to have maybe take 20 minutes to kind of have open Q&A, and then we'll kind of close up. I'll show a couple of slides about where you can find things. But now is the chance for all those questions that have been deferred or that were too deep. So maybe I can start with some of them. So one of the questions, let's see if I can pull this up. People are asking about CRISPR, which is something that's in the news quite a bit, and whether CRISPR is kind of, in the cards for ADNP syndrome. And I gave a kind of I typed an answer where I said that certainly, because CRISPR is a way of correcting genetic mutations, but I also said that I don't know if it's going to, I think it's probably a little bit far away, and there are other approaches that are a little bit more likely to happen in a more quick time frame. And so, and a related question that was asked was, if ADNP affects all of these downstream genes, shouldn't we be just focusing on, getting more ADNP rather than trying to figure out everything that's downstream of it? And so maybe I can start with an answer, with a partial answer, and then others can join in. So, CRISPR is an amazing technology. Again, it's revolutionized biology. Another Nobel Prize very quickly was awarded for that. It allows you to edit or to make changes in DNA. Making changes in a full-grown human being, of course, is still a long way off, right? Because it has to go into every cell and change every mutant DNA. And so and the technology is very, very much under development, and we're a ways away from that. However, the idea of targeting the gene or the gene product by any method is a very good one. So I think all of us would agree that if we could figure out how to correct the mutation as early in the process as possible, the better. But there are other ways to correct mutations besides CRISPR, right? So there are ways, for example, of targeting the RNA, right, which might be a little bit more accessible than DNA. There are ways of even targeting the protein, right? So, ADNP, as I said, there's you're missing a copy with some caveats that we spoke about earlier, whether there's a gain of function or a dominant negative effect. But if you're missing a copy of something, you still have a normal copy. So if you could figure out how to have less of the healthy ADNP degrade, or make more of that, or have less of the healthy RNA degrade, or make more of that, or fix the RNA that's mutated. Those are all things that are probably a little bit more accessible over the short term than CRISPR. But it's all the same idea. You have a mutation. Can you, if you can't fix the DNA, can you fix the message that it forms? If you can't fix the message, can you fix the protein? If you just don't have enough of it, can you stabilize what you do have so you effectively get more? Because everything in your body you make and you degrade, right? So if you decrease the degradation, you build more up. If you increase the amount of synthesis, you build more up. And so those are things that I think are in the works, and I think, I use the term gene-targeted therapy rather than gene therapy, which includes all of those things, including CRISPR. And some of those are much more likely to come into fruition in the near term than CRISPR, I would think. So there was a question about that, and I hope I kind of answered it. But Anna, you want to add to something about that?
Yes, I would just make a general comment that there is a lot of novel therapeutic modalities, and for better or worse, we all became familiar with things such as RNA vaccines. There is definitely a lot more tools available to drug developers these days, and we're trying to take a comprehensive look and really be agnostic in terms of selecting the tools and find the ones that best fit any specific disorder, any particular mutation at a more personalized level.
And I think that's a great point that I forgot to mention. Some of the things that people have been toying with, for example, getting getting mRNA vaccine, getting them into human beings, moved very, very quickly under COVID. And now, as you probably know, they're trying something similar for HIV, and all these things that might have taken a few more years are actually being deployed. And so there are ways now with nanoparticles and things like that to put things in the human body very safely. We know that because we've had millions and billions of people immunized now with this new technology. And so some of those methodologies are probably going to be a useful framework for some of the new gene-targeted therapeutics. Okay, what is the main goal of studying treatment options? Is it to cure or to manage symptoms? Alex is going to jump on that one.
I think cure is a very ambitious word. I think to manage symptoms, and hopefully the more robust the treatment, the more effectively we can manage the symptoms. When you talk about medicines like ketamine, clearly they're more likely to manage symptoms and maybe promote learning compared to medicines that will eventually be targeting genes more directly.
The terminology in drug discovery development circles is often disease modifying drugs. So no one really wants to talk about the cure because it's so difficult to define it and set the bar. But if you induce significant changes in the disease progression and the quality of life, that's considered to be disease modifying. And that's what we're striving for. And that can be in the domains of cognitive and behavioral aspects of the disorder, or perhaps even other areas that are more physiological than psychiatric.
There's a question here that Sandra could best answer, but we'll take it offline. So I have a question here about whether stem cells. So we've talked about induced stem cells. So induced stem cells mean stem cells that aren't natural, but we using Yamanaka's tricks, we take a blood cell, for example, and turn it back into a stem cell. But of course, there are also natural stem cells, and one of the sources from them is the umbilical cord. And so one of the questions is it worth saving stem cells from individuals with ADNP for research or for future treatment if there was a cell-based treatment? Anna, why don't you take that?
I'm sorry, could you please repeat that?
Is it worth saving cord blood or dental pulp stem cells?
I think it would be valuable to save any samples possible. If we talk about the cord blood for iPSC research, I don't think that that's necessarily something we need at this point, but I just generally think that saving cord blood would be a good idea for medical purposes. About dental pulp or any other samples, there is a number of tools being developed. There is a number of biomarkers being developed. We actually have a company spun off Mount Sinai that uses dental samples that has collaborated with Seaver in the past. Obviously, this work will require a lot more validation. But since we know relatively little about ADNP syndrome compared to how much we need to know, and despite all the progress that was made, I think it's not a bad idea to collect all the specimens that can reasonably be collected.
Let's see if we can get Sandra back onto the chat.
And I'll jump in to mention if anyone does have baby teeth, we are collecting those and can help facilitate that collection at Seaver.
But I don't think we're taking stem cells from that, right? It's just the teeth themselves.
Yeah, exactly.
Sandra, there you are. Hi.
I'm upstairs in the kitchen now giving Tony lunch.
Can we ask you to answer a couple of questions?
Yeah.
So one of them is the data in the registry available only to researchers or also to parents?
Oh, sorry. Yeah. Information, just aggregated information will go live anytime anyone's entering it. So 3 patients in there, you're going to see data on 3 people. 100, you're going to see data immediately on 100. And it's going to aggregate things like how many kids have developmental delays, how many kids have sleeping problems. It's going to just kind of collectively create a picture, and it will be instantaneously.
Thank you.
As well as we can always pull reports that families want to see.
Yeah.
But it will all be de-identified.
And then there's several questions about NAP, and I'll give a partial answer. So we will, we do plan once, if and when we see interesting phenotypes in the stem cells, which we certainly expect to, because I've already shown you that some of the collaborators are showing some things. We will try NAP in that system. But there are people I think some of the families are also asking about, what is the bigger plan with NAP? Maybe Sandra, you want to tackle that?
We are currently working right now with the NIH, and I was supposed to have a call yesterday with the FDA, but she had to cancel. We are still actively pursuing the use of it. We're just trying to go about it in a different way.
Thank you. So that, and then one of the questions maybe about the SAND is, how can a child be evaluated with the SAND, and can it be brought into routine use as part of ongoing care, ongoing evaluation?
Sure, yes. So I can, if anyone reaches out to me, or I can type in the answer box, the link for the SAND, if anyone's interested in sharing with their child's therapists or clinicians. And it certainly can be used in the context of treatment, both to identify sensory preferences and try to make a treatment plan that integrates your child's preferences, but also to look at change in symptoms over time.
And Paige, I sent you a message. If we have the ability to put the parent questionnaire,
It's
survey?
actually, yeah, you know what? It's actually a clinician administered interview. It's not a
okay.
measure. But thank you.
And then there's a question about ADNP and Alzheimer's disease. And quite honestly, I am not an expert, and the people who are experts are not on this panel. But I think people do know that there was an interesting study that suggested that tau pathology could be induced by ADNP haploinsufficiency, and I think that's an interesting finding, but it's still at its early stages, and I don't think we need to worry about Alzheimer's disease just yet. But having said that, if there is some kind of tau pathology, there's a whole massive group of top scientists all over the world and major companies that are trying to address tau pathology. And so if it is an issue in ADNP, we will benefit from all of that work to be able to address it without necessarily we can let the Alzheimer folks, experts, work on the tau issue. And there's a larger group of disorders that are called tauopathies, that there's a huge effort in trying to develop treatments for tauopathies. And if and when it's necessary, that is something that's being actively developed by really major groups, including pharma. Pharma obviously has a major interest in neurodegenerative disorders because of their widespread prevalence. Okay, and then nonsense mutations. I think there's a couple of complicated questions here, but There's also one very easy one, but
I'll take the easy one, Joe.
yeah. So we have a question about nonsense mutations. Are they harder to treat than missense? The answer is it depends on the gene, and with ADNP, we have yet to see a difference clinically in terms of nonsense in any kind of obvious way, be nonsense and missense, and even biochemically. Again, there may be differences mechanistically, but it's not like some of the what was mentioned here was Duchenne and cystic fibrosis. In those cases, some of the missense are what we call they're a light mutation. Right? They're not as deleterious as a nonsense. But in ADNP, the missense and the nonsense seem to be equivalently good or bad. Right? So I don't think right now we have any data that suggests that nonsense is better or worse than a missense. It's very different in Duchenne and even in some examples of cystic fibrosis, where the missense retains some of the function. So we don't think that's going on in ADNP. We don't have enough data yet, but so that's to Amanda's question. And Amanda also asks about we call it neurodevelopmental, but maybe a better way to put that is, what does that mean? As the families know, that some children and young adults have died with ADNP syndrome. It's like persists over life. Why do we call it neurodevelopmental, Alex?
Why do we call it neurodevelopmental? Because it first occurs in very, very early childhood, if not infancy.
Yeah.
It affects the brain, and it fundamentally alters developmental trajectories.
Yeah,
It's kind of a large basket term.
Yeah, exactly. Even if it's lifelong, right?
100%.
And talking about premature death, maybe Sandra can comment on this. Right? Many of these disorders can increase risk of premature death. Right? Seizures, for example, autism with seizures. Yeah. Sandra, you're on mute if you are.
Sorry. Most of the children who have sadly passed away have been due to some sort of viral infection or infection. I know there's great concern for one child right now, and it's all infection.
Yeah. So yeah, and so and of course, one of the things one wants to understand about these disorders that affect many systems is, are you more prone to infections? Are you more prone to, and how can we deal with that? And I think that in some ways those might be more, those are in some ways more tractable. Those are going back to the question of disease modifying versus treatment. If you knew that some disorder was associated with greater risk of infection, you might be able to treat that even if you don't modify the disease course. Right? I will note that Ilana posted, Ilana Gozes, and she has been participating. I've seen her name, and she said that I think what we all know, that she's working to develop NAP, and also doing a lot of work on the relationship between ADNP, NAP, and the tauopathies, which I just mentioned. And she'd be happy to talk at a later stage about all of this. So sounds like Sandra's wish is going to come true. There's going to be another day of learning. We'd like to have these at least annually, but you guys can tell us if you want to have them more frequently. What else can we, what else? Yeah, and then a question again about what we call genotype phenotype. Is there a relation between where the mutation occurs on the gene and the phenotype? And the answer is we haven't seen that yet. It doesn't mean it's not there, but it's certainly not obvious. Yeah, and also, so where the mutation is in the gene, we don't have any suggestion that it's better or worse yet. Of course, if we had 500 individuals well phenotyped, we could make a better statement, but nothing jumps out. Right? In some disorders, you see the first 10, you already see that the kind of mutation or the location of mutation seems to be driving a lot of the severity, but we don't see that yet in ADNP syndrome. All right, losing my voice.
Hey, Joe, you didn't see that in the methylation differences either, right?
Well, so they were the interesting thing was the methylation differences were big, but the clinical differences were not. Right. So, okay, and then a question we touched upon this, and then I think we can try to read and ADNP protein quantity, right? Can we change that? And that's as I said, there are companies that work on nothing other than how to if you have too little of a protein, how can you make more of it? Either by making more or degrading less. You know, the turnover of protein. It's like a sink. You know, the faucet is on. You could turn the faucet higher, or you can close the drain, and either way, you get more water in the sink, right? So a lot of companies are trying to figure out how to inhibit the natural breakdown of proteins, the natural turnover of proteins. So if you don't have enough, you get more simply because less of it's degraded. So that is what in that general category of gene-targeted therapies. And then a question about longitudinal studies about is the Seaver Center going to do longer-term follow-up? And that is, of course, something that's well worth doing. It's not for the faint of heart because it's a many years thing, but we are now beginning to think about longitudinal studies in different disorders. And one of the, there's a few good things about COVID. One benefit of COVID is everybody's dog has had somebody home for 2 years. And now those dogs are unable to be alone anymore. But the second benefit of COVID for us, as Paige mentioned, is that we did the CHAMP1 study was done entirely remotely. So we spent the first quite a few months of COVID developing tools and to do 80% of all that intense stuff that you've all done at the Seaver Center remotely. We can't do EEG, we can't do eye tracking, but we can do a lot of the assessment remotely. And so that does make an opportunity, makes it much simpler to do longitudinal studies because we don't have to bring families back, they don't have to fly in every year or so. So that's something we are open to, and we're happy to discuss with the Family Foundation. Sometimes it's best to do cross-sectional first, look at different ages, just to see what might change. First of all, it gives you data much more quickly. But of course, if there's any suspicion of change over time, you want to do longitudinal studies. And of course, some of the tools that are out there and some of the tools we're talking about, and even the tool Paige developed, can change with time. In other words, if things change, you can measure that change up or down. So here is a question from a new family, recent diagnosis, and that is a dramatic moment, and we share your feelings. Recent diagnosis, and wants to learn more. What's the best way to kind of ramp up quickly to understand knowledge, next steps, and support? And the answer to that is, of course, definitely join the foundation. They are really the go-to and a place where you can find other families and learn from them. And you can also reach out to us, email anybody on this panel, and we can talk to you. And if there are things we could communicate with clinicians, we're happy to do that too. So I'm not naming names here, but except Sandra. Go after Sandra. But I would definitely join the ADNP Kids Research Foundation without question, and the Facebook group, and get all the mailings. But you're also you can reach out to us, and Sandra probably could give you other expert sites that might be closer to home if needs be. So I think All right, I'm going to just show the last couple of slides for the wrap up. I can, if I can just find them. Yeah, here we go. So we have these collaborations and actually a few more that we haven't talked about yet. So first, I want to thank everybody for attending. Like I said, it's great for us to interact with families. It's unfortunate it's via Zoom and via chat. A long time ago, we tried to do these things in person, and we actually did them in person historically. And nowadays, it's we're not doing it that way. But maybe we'll go back. Although, of course, doing it remotely has the advantage that anybody in the world can join. And as you know, we have people from all over the world on this call. It has been recorded, as you probably saw. And this is our website, seaverautismcenter.org. It takes a little bit of time to get it up there. We kind of go over it. We take off any tailing things, but then it's posted. So it'll be available on our website. You can watch it many times as you'd like to. Many of you probably have gotten emails from Rachel Cohen. So please reach out to her if you have any additional questions. And, you know, a plug for the Seaver Center, not to take away from the Family Foundation, but Seaver Center does rely heavily on philanthropy. We have a lot of grant support from NIH and from private foundations and public foundations, but we also we have the flexibility to start new projects even without funding because of philanthropy. And within the ADNP sphere, we probably talked about 5 new things that we'd love to do, which we'd be happy to do, but it often comes down to money. As you probably know, in the research space, none of this is covered by the hospital. It's all covered by what we call soft money. So when a family comes into the Seaver Center for a clinical research assessment, that is not covered by insurance. It's not covered by the hospital. It's covered by funds we raise. And we do apply to NIH for ADNP money. Alex has one in, Nan and I have one in. Takes a while because it's a rare disorder and it hasn't really quite filtered up how important it is. But one of the things some people have done is if they've joined the associates board, and actually there's an ADNP family on the board, and that kind of helps us have kind of one-on-one feedback on our direction. So that's something that you could consider. Sarah Lynch is the person to communicate to about both of these things, and her email is there. And then with that, I'll just say thank you again, and it's a real privilege to work with the families. It's important. We are all passionate about trying to be helpful and seeing that our research makes a difference. And I think I know from the family's perspective, we're barely moving, but from the point of view of how these things work, as Alex said, the quickest trial ever, and we got, as soon as Nan Yang came to Mount Sinai, we roped her into the stem cell work, and so we are. It is a pleasure to work with all of you, and we appreciate all your support. And yeah, I'm going to stop rambling. There, I've stopped rambling. Sandra, anything you want to add? Alex, Anna, Paige?
Well said, Joe.
Nothing. Okay.
Well.
Bye.